In my talk today, I'll briefly describe some of our highlights of our study about the neural substrate for animals' innate behaviors.
Basal forebrain is a complex nuclei located in the ventral part of the forebrain area, containing different types of neurons. Our lab is interested in
cells are identified to play a role in neutrophilic asthma. For example, when bacterial infection, air pollution stimulate lung mucosa.
Hello everyone, I'm Alice, and my topic is focused on a novel protein JHP0417 and try to investigate its role in
heavier levels of these mice. The neurological function in sensory motor getting is impaired in patients with schizophrenia.
ELA and sucrose gradient experiment to identify dynein FOXP2 knockout cells. The overexpression of dynein-1 could lead to the collapse
where the plasma membrane will eventually rupture and release the net component. So first, we will
the nucleoside transporter that located on the membrane of lysosome and mitochondria.
In animals' brain, the adhesion molecule is very important for synapse formation, and citracone is one of them.
In cell initiation, there are two critical proteins C1C4 will recruit TPK2 on the centrosome and promote CR formation.
Imagine yourself swimming in beers and wines for up to 15% of alcohol for about a month.
Double kills can predict reward in training stage. In every 2 seconds, one of the like kill and one of the cell kill will present simultaneously. So there's
whether or not you have inherited epigenetic marks from your parents, grandparents, or even great-grandparents. Maybe you do or maybe you don't.
Focus on the amygdala to the dorsal striatum. And first, we inject two retrograde tracer into the dorsal and the ventral.
Degenerative disease with a symptom of memory loss. Even though every 3 seconds someone will develop dementia, there's still no effective
elevated increased level in patient of the chronic migraine. And throughout the whole brains, we found pinpointed specifically in the central region of
protein with a conserved carbohydrate recognition domain CRD. And glytin-3 is the only chimeric type in glytin family.
Sounds great when we are talking about the food, right? But when we are discussing about our body composition, it is not happy to see too much fat accumulate.
Can rarely infect epithelial cells, so it makes it impossible and very difficult to study EBV infections in epithelial cells.
We go treat to ask for sweet, but not only does the children love sweet, so does adult like me. Yeah, we drink bubble milk tea.
90% of cases with mutation or overactivation. Therefore, afatinib, this is an EGFR inhibitor, is expected to be an effective treatment
in the lymphoid cell, ILC2. So what is ILC2? There are 19 non-beam lymphocytes, and which can be activated by our
regulum panel. Here we use in the human whole blood sample. And indeed, our RS regulum is enriched in different levels in the immune cell types.
To the LHB, we hypothesize that they may be a part of the aversion system and may oppose the real processing through the LHB.
UPT5. They form a complex named DSIF and interact with RNA polymerase II.
It's in the immunocompromised patients, so that's why we target on this disease. Okay, and the uropathogenesis is composite.
Rule prediction error, or RPE, is the discrepancy between actual and predicted reward value, which has long
not simple as those we handle in vitro. Moreover, because the adhesion signal will have spatial separation, so